In my presentation, I've erred on the side of pointing out pitfalls, and I also probably have more stuff about histopathology things in this post-mortem analysis stuff. And I've also spent some time talking with David Meyerholz here, who's our resident comparative pathologist that's very involved in our studies. Let's see here, so let's do this. So these were the topics we were going to touch on. You know, as far as I missed the part where you guys talked about some of the virology. So, you know, just running down a few things of what we've been doing. You know, been working with the Washington strain. We've sequence verified it. And we want to look at viral RNA expression. We've got primer sets for genomic and subgenomic RNA. And, you know, we've used PCR to look at that as outlined here. We've also titered tissues, multiple tissues. I have an example of that on the next slide. And this is pretty much, you know, the methods that we've used. We're also, you know, doing virus evolution, and we've been working with our clinical colleagues looking at some cohorts of infection. And we've been using the Oxford nanopore technology and following this Arctic network protocols for the sequencing work. You guys are probably familiar with this, but it's like a living protocol that people all over the world are using. It's a very handy way to do whole genome sequencing of the virus. And of course, you can also do PCR to amplify across regions of interest using Sanger sequencing or what have you. And we've done that to look for deletion of the furin cleavage site based on passage in tissue culture, for example. I think, you know, some pitfalls to think about related to this part of the talk is, you know, being confident that you can isolate high quality RNA, keeping in mind that there might be slough cells either in tissue culture or animal models that could influence your results. If there is a viremia in your animal model, you know, you may have low level RNA detection in multiple tissues, but when you look for virus by titer, you may not see anything. And, you know, if you're doing serial passage in an animal model, you know, there may be a point where you want to plaque purify certain isolates and characterize them in more detail by sequencing and animal studies with those isolates. Do we want to talk about things slide by slide or run through the whole thing?
I think run through the whole thing and then we'll get back to.
Sure. So, I mean, this is just some data from our recent work with the K18 mice showing on the bottom here, looking for virus by qPCR for the genome and then titering across multiple tissues here. We've also been playing around with nasal lavage in mice to recover virus. And what we initially tried was lavaging the nose with 20 microliters of saline, but the recovery was minimal. So leveraging something that we know about airway function, we treated the mice with a cholinergic agonist to stimulate secretions from submucosal glands. And that worked quite nicely, so we could, you know, get a gush of fluid that allowed us to collect the secretions and show that we could detect virus in that, which we quantified by PCR as a CT value over a volume of lavage. And with the histopath analysis, having done a lot of things with animal models over the years, having a a board certified pathologist, especially one that happens to be trained in veterinary medicine and animal models, is a huge plus. I think as you analyze tissues, it's really important to keep in mind what we know about the human disease as we try to look for parallels in animal tissues. And that's where, having somebody on the pathology side who's knowledgeable to consult with and apply their eyes is quite helpful. And it's also important when you're getting help from that pathologist to interpret experimental results that you use a masking approach to avoid bias. So, at the sort of gross pathology level, there may be reasons to look at lung tissue to complement or other tissues to, I'm kind of lung centric, but this should be generalizable to any tissues. There may be settings where it's important to photograph and score tissues either before removing them from the animal, or if you remove like the heart and lungs in block, for example. But it's also important when you're doing that to remember that if you need to do this prior to fixation, or otherwise you'll change everything you're trying to look at in terms of the color and surface texture, etc. So just running through some nuts and bolts of what we do for histochemistry. We usually use 10% neutral buffered formalin or 4% paraformaldehyde. Depending upon what you want to do, you might want to use other fixatives. Using the right ratio of fixative to tissue is important, and also it's important not to have really large pieces of tissue that you're trying to fix, or otherwise you won't properly fix them. With lung tissue, we generally try to do an inflation fixation and also perfuse the tissue to get the best morphology and not have artifactual atelectasis. In some settings, you may need to do cryopreservation, and that doesn't always give you the best morphology, but it might be necessary for example, for certain immunostaining techniques that won't work on on paraffin embedded tissues. So we would commonly use H&E staining in our assessment. We've been using the N protein as a target to localize antigen in tissues. We've tried several antibodies. This one that's commercially available seems to work quite well in our hands under these conditions that I've outlined here. Just an example of that here, where we have an uninfected lung here on the upper left, and at 2 and 6 days post-infection with 10 to the 5th PFU in the K18 mouse model, we see this diffuse antigen presentation in the parenchymal tissue and also in the airways. Also in some animals in the K18 model, late in the course of infection, we can see diffuse central nervous system infection. So in terms of some things to think about with pitfalls, a lot of careful planning is helpful. You want to make sure that you're using high quality tissues. So harvesting them as rapidly as possible to minimize autolysis is important. You can get a lot of artifactual changes otherwise. There may be considerations related to what strain of mouse you're working with that could influence infection susceptibility or severity, as age and sex can also similarly influence things. Another thing, with looking at lung tissue in particular, is that the method of euthanasia could influence the outcome. So for example, inhalational agents can introduce edema and hemorrhage. So, you know, having the appropriate controls is important, or you might want to select an intravenous agent. Also, you know, unlike other organisms, the lung has dynamic size changes during respiration. So how you handle this post-mortem to avoid atelectasis, you know, is another consideration to get proper tissue preservation for comparisons. And a common thing is to use right ventricular perfusion of a fixative into the lungs prior to removing them, and also to insufflate the air spaces in some settings. So those are all just things to take into consideration. You want to have the appropriate controls. And you want, you know, when you write a paper about this, it's helpful if you carefully detail how you did these things. So that's another point. I guess the other, you know, thing that's helpful if you're trying to make comparisons to human disease is knowing some of the differences between the animal models and keeping those in mind. For example, you know, mice have different airway cell anatomy. They have different configuration of their lung lobes, with 4 right lung lobes and only 1 on the left, while humans, as you all know, have 3 on the right and 2 on the left. And then another thing that I think adds rigor to your evaluation of the outcome of your studies in these models is to do some quantitative assessment of the histopathological changes. So that, you know, what you're looking for are different kinds of lesions that occur. And in the lung tissue example, in response to coronavirus, severe coronavirus infection, diffuse alveolar damage is a common finding. And I've listed here a number of the common outcomes that you might see associated with such an infection. And these are all things that can be scored as you evaluate tissues in a quantitative manner. So a common way to do this is to do semi-quantitative approaches where you have a scoring system that you decide in advance and apply to the lesions that you're trying to score. So I've listed an example here from some recent work we did with the K18 mice, where, for example, if we're looking at edema, hyaline membrane formation, and necrotic debris, the scoring is looking at 200x magnification and then quantifying how often these things are appearing in those fields, and so on, for neutrophil infiltration or other types of lesions you might think are important to note. And, you know, this is a cartoon from one of Dave's publications, you know, just using an example of, say, pulmonary edema on the top, where this is a bronchiole and these are air spaces, excuse me. And you know, if there's alveolar pulmonary edema, you see these spaces start to fill up. And that contrasts with what you would be looking for with hyaline membranes, where these red lines are showing that type of change in the alveolar region. And so this shows an example from the K18 mice where there's diffuse alveolar damage over the course of the infection. It may be kind of hard to see, but these asterisks are over areas where there's edema or hyaline membrane formation. There's a variety of cellular infiltrates and debris and dying cells. And then on the right here is an example of some histopathological scoring at 4 and 6 days post-infection. And there are also, you know, other scoring mechanisms, quantitative scoring. You know, for some applications, it might be more precise and sensitive. You know, typically there you might be taking very high quality photomicrographs and use software to analyze the tissues. And, you know, there's a bunch of considerations for what's the right method for your particular application. And the last thing I had was just looking at host responses or cytokines. This could be done in tissues or specific cell types. You could evaluate it at the transcript level with RNA-seq or single-cell RNA sequencing, RT-PCR. I mentioned some primer sets at the end of this. There are also some commercial platforms that have gene sets to look at this. And at the protein level, you know, you could use ELISAs or Luminex panels, or NanoString is another commercial panel. I haven't used these, but I've certainly seen them used in several publications, and maybe others can comment on that. And this is just an example of using qRT-PCR to profile a number of host response genes of interest at different days post-infection. And then I put I put some histopath references and a cytokine PCR reference at the end. So that's what I have to run through.